Hello, welcome. I'm Dr. Tony Magliocco, the Cancer Informant. Today's episode will be focused on clinical trials. What are they, and should I consider one? Now, what are clinical trials? There's a lot of confusion about clinical trials, and many patients are under the impressions that either they're very experimental or else they're really only an option for late-stage cancer patients who have lost all hope of any other sorts of treatment. This could not be further from the truth. In fact, clinical trials are part of routine care, and in fact, they should probably be available for all cancer patients. Now, let's take a step back and consider what exactly is a clinical trial. Well, when we think about clinical trials, and particularly interventional trials, this is where we think about taking a new treatment approach for a cancer patient to determine if it's an advance or more effective than an existing treatment approach. Now, treatments could really be anything. They could range from doing nothing, for example, just observing a disease progressing and waiting to stage an intervention until it has achieved a certain progression point. It could involve surgery. It could involve using different types of systemic therapeutics, for example, chemotherapy or targeted therapies. It could involve special types of immunomodulation, checkpoint inhibitors, or CAR T therapy. It could involve special types of external therapies, for example, radiation with photons, with electrons, with other types of physical energy, magnetic waves, heat, cold, etc. Could even involve biologicals, for example, viruses, altered cells, and so on. So there are many, many opportunities for therapeutic intervention for a cancer patient. Now, generally, we consider a pathway or a timeline for the discovery of therapeutics all the way through to practice change. So most therapeutics begin their life as an idea in the mind of a scientist or an investigator, determining, is this new agent potentially effective for cancer therapy? So often they begin their life in a university setting, perhaps studying the impact of a new molecule or a treatment approach on a cell line or a tissue culture. This is where cancer cells are grown in a test tube, and we determine if the impact of a therapeutic has some sort of change. If a therapeutic appears promising, which means that it will kill cancer cells, but spare normal cells, it can graduate to the next level. This is where more complex systems are utilized. For example, organoids, where groups of cells are grown, or tissue cultures where there are a mixture of cells. If it appears that the agent or technique still works in that particular system, we tend to move it to a preclinical phase. In this phase, the agent is exposed to typically animals, perhaps a mouse model or some other type of vertebrate model, where we determine, is this treatment safe, first and foremost? And secondly, does it appear to have some sort of effectiveness? Does it really kill cancer cells? And typically, we would also compare it with some standard treatments because we would want a new treatment to be at least equivalent or better than an existing treatment. These are all the preclinical phases. And at this point, we can make it a determination. Does this treatment appear to have effect? We can also determine what sort of cancer does it affect on, and is it subtypes of that particular cancer? So we really need to think about how might this treatment be utilized in a clinical setting? Is it in a subset of cancer? Is it got to have a certain type of mutation or alteration? Will it only work at a certain stage of disease? And so on. So this information is important then when designing the clinical implementation of the study. And there are typically 3 phases to clinical implementation: phase 1, 2, and 3. Now, phase 1 clinical trials, the purpose of a phase 1 trial is not to determine the effectiveness of a treatment, but really to determine if a treatment could potentially be harmful. Because we certainly do not want to give a treatment that will cause toxicity or unexpected side effects, which can be terrible or even lead to death. Determining the safety profile of a new therapeutic is paramount. Typically, phase 1 studies may even be used on normal people or volunteers, often medical students or other types of people who may volunteer to be exposed to a particular treatment to determine if they have toxic side effects. A phase 1 study will generally only have less than 10 people or at most less than 20. And essentially, an escalating dose of the treatment is utilized to determine at what level does toxicity occur. Now, obviously, phase 1 studies need to be done in carefully controlled environments so that toxicity can be monitored. And often a lot of additional studies are done alongside of phase 1. For example, blood samples are obtained, blood measurements are obtained to measure is the compound accumulating in the blood. There's evaluations of cardiac and lung function, brain function, etc., to be certain that this new type of treatment is safe, and also to determine what is the therapeutic level, at which dose does this treatment become potentially toxic. So essentially, that's the phase 1 studies. And there are many phase 1 studies across the country. Now, in a phase 1, it's also considered what we call first in mankind. And this is where a cancer patient may have the opportunity to first get dosed with an innovative treatment. Now, the downside of a phase 1 is that a dose may be suboptimal or too low to actually have an effect. So when utilized on a cancer patient, it may or may not be effective. So some phase 1 studies are utilized on cancer patients, but one must remember the purpose of a phase 1 is toxicity, not efficacy. Efficacy means response to the therapy. Now, moving into phase 2 is where things get much more interesting. This is where we really begin to look at, is this agent actually making a difference? So a typical phase 2 study will have many more people. So generally maybe as low as 20 or 40 or as high as 100 or more. And this study is designed to identify something called a signal. Is this treatment actually making an impact between two groups? And typically in a phase 2 study, there'll be two arms. So in one arm, there would be the standard treatment. And in the second arm, there'd be the standard treatment plus the new treatment. So the new treatment would be added. And often these studies are done in a double-blinded way, meaning that the person administering the therapy does not know which patients are getting it, that some of the patients are getting a placebo and other ones are getting the treatment, and so on. And the study may also be done in a crossover way as well, so that at some point in the study, the treatment arms may be switched so that everybody can get a dose of the treatment to see the effect. Now, there is also stopping rules in these studies, meaning if there is an unexpected toxicity, a pause will be taken in the study to determine, is the toxic effect something to do with the treatment, and should we reevaluate the dosing, or even whether this study is feasible. Second, there's a futility point where we look at the data at some point of the study to determine, does there actually appear to be effect in one of the arms where the treatment is being given? And if it doesn't appear to be there, should we stop this study so that we can actually give patients opportunities to go on to other treatments that might be more effective? These are various interim endpoints that are used for safety. So the reason I bring this up is to assure patients that clinical trials are extremely well thought out. They're backed by a lot of preclinical data. They're conducted by experts from around the country. Often they're done at multiple sites. And there's actually very, very close scrutiny of them by numerous regulatory authorities. This often includes IRBs, or investigational review boards, that basically look at, are these studies safe? How are they going to be monitored? What are the stopping rules? Also, the FDA may play a role to determine, are these studies being properly constructed? Are patients being enrolled correctly? Is the data blinded? Is there any bias in the study in terms of the investigators? Are there any undue influences that might encourage them to bias the data or enrollment? There is very, very strict scrutiny on these types of studies, and these studies are actually very expensive to conduct. They're always published as well. So you will see something called clinicaltrials.gov, where you can look and see all of the studies that are open, which ones are ongoing, which ones are closed, which ones are accruing. There'll also be interim results. So if there's a publication or an abstract, the information will be there for you to review as well. It's also become important that all studies actually publish, whether the results are negative or not. We're not really doomed to repeat failed studies over and over again that we can see, oh, this study failed. We are not biased just by looking at positive studies. So knowing the results, good or bad, from a study is important to determine how can we learn from this and how can we make future studies better. Also, it's important to know for the patients that if there's a very strong signal in the study, wow, these patients are doing extremely well, the study will actually be stopped And generally, the patients in the non-experimental arm will be shifted over to the new arm so that they can also benefit from these types of treatments. The phase 3 studies are particularly important. These are what we consider practice-changing studies, and this is where a very large-scale study is done to really get an FDA approval or approvals of treatment opportunities globally. So typically, these studies may have several hundred patients enrolled in them. They may come from a variety of countries to enroll patients, and they're done at a very large scale. Obviously, they're extremely expensive and very important to run and complete effectively. These are high-profile studies where patients are closely scrutinized. The data is often kept secret until an important disclosure event occurs, where they're usually presented either an interim or final analysis at some of the world's leading cancer conferences. These include meetings such as ASCO, the American Society of Clinical Oncology, which has an annual meeting, ESMO, the European counterpart, and specialty meetings such as the San Antonio Breast Cancer Meeting or other types of meetings where these clinical trial results can be released and where investors and others can look at the performance of a company and doctors can make an opinion about the effectiveness of these new therapeutics. If a study survives a phase 3 and gets approval by the FDA, in the United States, it then becomes available for marketing and for utilization. And that's where therapeutics can now be generally made available to the public. It doesn't stop there, though. In other countries, other formulatory boards will consider new therapeutics and whether they should be added to the formulatory, and more importantly, should they be covered. So many of the new therapeutics may not actually be paid for because they're deemed to be very expensive. So there's a complex process in terms of both running clinical trials and actually implementing them into clinical use that involves numerous regulatory authorities and also paying authorities and so on. After a drug has gone past a phase 3 has been implemented, there's something called a phase 4 analysis or post-market analysis. This is where a company or authorities may look at a new therapeutic to determine how is it being utilized in the general market, what is its impact in real-world use, and were there any unexpected toxicities or findings that may be of concern about the performance of a new therapeutic. This discussion is really to assure patients that clinical trials are a very, very well-established process. They are carefully monitored. There is a lot of regulatory. I, in fact, encourage almost every patient to look at the opportunities of participating in a clinical trial. There is also advantages to participating in a clinical trial in that there may not be a cost for participating, and the sponsors of a trial may actually help you travel to sites where the trial is open to allow you to gain access. What does it take to qualify for a clinical trial? Well, first of all, you need to meet the eligibility criteria. So each clinical trial has specific eligibility criteria. It might be that you have to have a certain type of cancer. It might be that the cancer has to be a certain stage, for example, stage 2 or stage 3. Perhaps there's a criteria that you must have failed a previous line of therapy. There might also be exclusion criteria that you might be excluded should you have been treated with a certain line of therapy or if the disease is in a certain state. There are other exclusion criteria that really affects the individual. What is their general state of health? So if a person is not in a very good state of health, for example, if they cannot walk or if they're short of breath or if the disease has progressed to a point where they're suffering a lot from progressive cancer, then they may be no longer eligible for a clinical trial. So if you're interested in clinical trials, it's actually important that you be in a position where you're well enough to qualify for a clinical trial. The qualification involves applying to an oncologist or center where the clinical trial is open, and the oncologist or the trial's intake group will have an assessment to determine if they have a position open for a particular individual and whether they qualify. Now, it might surprise you to know that there are actually thousands of clinical trials open. And in fact, most clinical trials fail because they fail to acquire and enroll enough people. And this is because of a couple of factors that people may not be aware of the trial. Doctors may not know the trial is open or remember to enroll patients. Patients may not have had the correct testing done to coordinate with entry onto the trials and so on. So unfortunately, most clinical trials fail to meet their endpoints simply due to lack of patient enrollment. And one of my goals as a practitioner in oncology and precision oncology is to try to make trials more easily searchable and available for patients so that they can consider this opportunity. If you're interested in learning more about clinical trials, please contact me directly. In summary, clinical trials are an important treatment opportunity for cancer patients. There are different phases of clinical trials, phase 1, 2, and 3. Phase 1 are for measuring toxicity and may not really be of general interest to a particular cancer patient. Phase 2 are probably more interesting to cancer patients in that they are now testing agents that have proven efficacy in early stages, and this is where access to novel agents can be accessed. Phase 3 are the larger multinational studies that are also potentially of value to patients suffering with cancer. You can learn about clinical trials through clinicaltrials.gov or through your own cancer center, or even asking your own oncologist about clinical trial opportunities. But remember, there are so many clinical trials available that your own oncologist or cancer center may be unaware of the very many treatment opportunities that are available. Hence, I encourage you to seek additional help in searching for clinical trials where you might be eligible for if you think you are interested in them. Also remember that you need to be eligible for them in that you do need to have enough strength and health to be able to participate in a clinical trial. So I hope that this discussion of clinical trials was useful for you. Again, please visit our website to hear other episodes of the Cancer Informant, and I welcome you to leave comments and questions. Thank you very much for your listenership, and I'm Dr. Tony Magliocco, the Cancer Informant. Have a wonderful day. Take care. Bye-bye.